Prodyot Banerjee. He is Bioinformatics CRO scientist in Biotechnica, Bengaluru. He will be guiding us on the topic exploring bioinformatics databases under the guidance of Dr. Bhavana Prasher, Senior Principal Scientist, CSIR-IGIB, New Delhi. Over to you, Pradyotji.
Okay, thank you, sir, for the brief introduction. So I really appreciate your energy in introducing me, and I just wanted to thank the organizers of this particular lecture series, that is All India Institute of Ayurveda Goa and IGIB Delhi. I also want to extend my sincere gratitude to Dr. Bhavana Prasher ma'am for giving me this particular opportunity to speak in this particular forum. So that is about me. So actually, I have done a MTech in Bioinformatics from Delhi Technological University, and currently I'm employed at Biotechnica Bangalore, where I am a bioinformatics specialist. So handling multi-omics data analysis and those kind of research, translational research I'm into. Apart from that, I my specialization is in bioinformatics and artificial intelligence. Yeah. So that's about me. Uh, so yeah. So without further ado, so can we get started, or should we be waiting for some minutes?
I think you can get started, Pradyot.
Yes,
Okay.
we
Pradyot
can
also
start.
worked with us in IGIB for some time, and
Yes.
that's how we know him, and he has been in touch also. So yeah,
Yes.
you can start, Pradyot. Your topic is very vast, so we have to also be in time.
Sure. Thank you.
So
Thank you.
Pradyot will be taking a little bit of hands-on. I mean, he will take a live demonstration also. So if any of you can sit in front of your laptops, it will be useful. So that is one thing I think I should tell, so that whatever links he will tell, you can take that, and also show how some of the searches that we can perform on the web resources. If anybody has a laptop or a tab or anything, even on the phone one can start, but that's something he's going to take in this session. This is going to be slightly hands-on for a live demonstration session.
Yes, ma'am. Yeah. Thank you, ma'am, for the brief. So yeah, exactly that is what we are gonna do. So basically, I will just brief you on all of the things in the theoretical aspect of the things in just 15 to 20 minutes, and post that we'll be diving deep into the practical aspect of the things, and we'll be going on to the demonstration. So side by side, you can start alongside me using your laptops or mobiles. Mostly, I would recommend laptop, because all of the web servers that are there might not open in your mobile, and if you're using a mobile, so in that go to desktop view. Okay, so yeah, so let's get started. So basically the topic for today is exploring bioinformatic databases. So we will look into the different databases that are concerning to multi-omics data analysis, to be precise. We want to study or like a disorder. Let's suppose we are taking a particular disorder, and likewise we can study that at multiple levels. Okay, so the first level being your genome. Okay, so because if you are building, let's suppose if you're constructing a wall, so the main thing that you start with are the bricks. Okay, so genetics are the bricks to basically what we are going to understand the etiology of the disorder in the further part of the things. Okay, so that is why we should start with genomics. That is the importance of genomics for you. But how this particular bioinformatic databases can help us to either go from disease or disorder to finding the genes responsible for that, or the vice versa. We can also go from gene to the identifying which particular disorder it causes. Okay, so with that particular note, let's get started. So I have already introduced myself. Okay, so let's get started. Yeah. So first things first. So what we are gonna see is like the different types of databases concerning to your genomics. Okay, so just before discussing the databases, so let me tell you one thing, that why we are studying this particular databases, and what is the relevance of understanding all of these things. So First, everything started with your human genome project, basically. I think those particular lectures you have already been a part of. So basically, with human genome, we were capable to understand that what is the reference genome, and based on that, we can now look at our query sequence and align our sequence to a reference genome, and we can see that. Like, let's suppose this is the reference genome which is having what it is having, basically the ATGC sequences. Okay, so how the particular ATGC sequences are arranged, because we have a 3.2 billion base pair genome. Okay, so how the things are arranged, based on that we will be knowing. Let's suppose I am having a patient. Okay, so the genome of that particular patient is over here. So how would I know that there is a mutation? Okay, so if I see that here it is a where it should have been a T, so that is the mutation for you. Okay, so that is one kind of a mutation, a single nucleotide mutation, but there can be multiple types of mutation that are there. Okay, because we will be talking about variants, so that is why I'm just giving you a like brief that. So basically, the initial first generation of sequencers were there. So when we talk about Human Genome Project, we had our Sanger sequencing and all. Okay, that happened. Using Sanger sequencing, it took almost 32 years to be precise. If we are talking about reference genome, okay, so it started in the year 1990, and it is just only in the year 2022 the whole genome got covered. Of course, the Human Genome Project was released in the year 2003, but that was having only 75 to 8 like 85 to 9 like 85% coverage, almost 75 to 85% coverage it was having back then. So it is just in 2022 it got covered. So it was obviously a slow method. So but that is what was available back then. Okay, so after that we have our next gen sequencing, that is the second generation of sequencers that we have nowadays. After that we also have third generation of sequencers, like from the likes of your Oxford Nanopore and PacBio and likewise. Okay, so we have so fast in the genomic context. Okay, so we have tried to rationalize the cost, bring down the cost by a fair margin. Okay, and now we can do all of this job in one day. So that is the transition. I just wanted to give you a picture of what is the transition. So now we can basically sequence the whole genome of an individual in one day also. So which took this many years initially. Okay, so because of the advent of this particular thing that is called as a reference genome. And remember this particular thing, reference genome, because we'll be talking a whole lot about this. Okay, so in all of our databases, because all of the databases we will be like checking our query sequence to the reference genome, aligning it to the reference genome, then we will see what is the variance, and based on that we'll be identifying what is the SNP, that is a single nucleotide polymorphism, one kind of variance again. Okay, so what is single nucleotide polymorphism? So basically single nucleotide base change that is repeated over the genome multiple times. Polymorphism means, poly means many, morphism means repeats. So multiple time repeated over the genome a particular single nucleotide variance. Okay, because we are going to decide discuss a database on that as well. Okay, so that is about the brief on all of the things. Now let's come on to the databases. Why do we require databases? What is the relevance of it, and how the data are stored in this particular databases, where it is getting those data from. Okay, so there must be some experiments that are happening, and based on what we are having the data sets. Okay, so all of the cohorts that are there in this world, apart from that, all of the studies that happens. Okay, so from the likes of your that's happening in your EBI, that is European Bioinformatics Institute. So that has a headquarter in UK. So then you have in the US you have NCBI. So I will be running you through NCBI. All of these are primary databases. So we'll also be discussing about what is a primary database, what is a secondary database, why do we require one. Okay. So with that particular note, let's get started with the first type of databases, that is your genotypic and phenotypic databases. Okay, and how this particular thing can give us a picture of the clinical relevance of a particular variance. Because as you know, all of the variants are not going to cause a disorder. There might be some silent variances and silent mutation that will not reciprocate to the disordered condition. Okay, so that is why we need to see. So first in that is your GWAS catalog. Okay, GWAS basically stand for your genome-wide association study. So it is a very famous study which intent to basically identify the pool of SNPs, single nucleotide polymorphism changes, okay, across the globe. Okay, across the globe, what are the common SNPs causing a disorder and what are the different SNPs causing a disorder? Let's suppose I take a cohort from America and I take a cohort from India and studying the same disorder, that is breast cancer, let's suppose. Okay, so there would be certain certain SNPs causing the breast cancer in both of the locations. Okay, and there would be certain SNPs which are particular to a geographical location. Why? Because genomics is basically influenced by multiple factors. Okay, one of them is your lifestyle. Apart from that, your location. Okay, so all of this basically governs genomics. So that is why we need to understand that. And with those particular study-based data, so the worldwide that particular study has happened, and based on that, we have that particular data, and that is stored under a database called as your GWAS catalog. So we can leverage this particular leverage the use of this particular database to see that what is the association of a particular like a disorder and what is the link of a particular genetic variants with the disorders, and likewise I will show you everything will be demonstrated. Okay, so I'm just giving you the a brief introduction of all of those things. Okay, then we will move on to the demonstration. The second thing is your FeeGenI. So FeeGenI basically is an integrated database which can give you an idea of genotypic and phenotypic changes. Okay, and how it revolves around a gene, or you can give also the set of gene or pool of gene. It is not that only one gene you can search using. Okay, multiple genes you can attach, and you can see what is the relation between all of the different genes connecting to that particular disorder. Okay, that is also possible. Apart from that, it also gives you the clinical significance. Okay, another database is also there which gives you clinical significance, just to tie the strings between the databases. So FeeGenI is there, and apart from that, you have ClinVar. I will be talking about ClinVar in a while. So ClinVar is another database which will give you clinical relevance. What do we understand by clinical relevance? Clinical relevance basically means a particular variance is going to cause a disorder. What are the likelihoods of that? How much likely it will cause a disorder or not? Okay, so there are multiple categories that like pathogenic, then your benign, pathogenic, uncertain. So likewise, what is the probability of that particular variance to cause a disorder? Okay, so in that particular lines, we have these two databases majorly. There are a whole lot, but majorly we look for these two databases. Then if we are concerned with knowing of a disease condition or disordered condition, okay, so we can head on to OMIM, that is your online Mendelian inheritance in men. That is the full form of OMIM. So here we can go and search by two methods. Okay, so one method is obviously gene to disorder, like we can search by the gene term and it will redirect me to the disorder that it concerns to or associated with. And we can also do the vice versa, that we search by the name of the disorder, let's suppose Parkinson's, Huntington, or type 2 diabetes, mellitus, and likewise. Okay, so we can search by via that, and it will give me the pool of gene or the genes that has been studied with evidence. Okay, so with the concerned paper, that how this particular gene have their role in causing that particular disorder. Okay, so that is there. So that is what we can infer from your OMIM. Then comes your ClinGen. Okay, so ClinGen again is a clinical relevance database. Okay, so which can be used for precision medicine. Although for precision medicine, all of the genetic databases can be taken into account. Okay, so it depends upon what kind of a precision medicine or personalized medicine you want to come up with. Okay, so now coming on to your key genomic and variance databases. So there are many. Okay, but we will always start with NCBI. So that is the primary database. Okay, so what is a primary database? Basically, in terms of bioinformatics, a primary database is a database which stores all the raw data from the experimental from the experiments basically. Okay, so whatever experiments are happening worldwide, or studies are also involved in that. Studies, experiment, all of those things, the raw data is stored in this particular databases. Those are called as your primary databases. Majorly, we have three primary databases. One is called as your NCBI, National Center for Biotechnology Information or Bioinformatics Information. Okay. So then you have your EBI, EMBL. EBI is European Bioinformatics Institute. Okay, then you have DDBJ. Okay, so DDBJ, these three are the majorly used primary databases in bioinformatics. DDBJ stands for your DNA Data Bank of Japan. So it is based out of Japan. EBI is European, it is handled by European subcontinents and having the headquarters in UK, United Kingdoms. NCBI is handled by US, okay, so handled by US government. Okay, so this is from US, this is from UK, and this is from Japan. Okay, so as I have already stated, they store the primary raw data from the experimental things. Okay, so GenBank, then coming on to the GenBank is part of NCBI. Okay, so under NCBI there comes many tools. Okay, so I will show you the NCBI UI. In that you have a drop down menu at the top. So in that you have plethora of options. Okay, so one of them is GenBank. Okay, so then you have your gene expression omnibus also comes under that. Gene ontology also comes under that. Then your DBSNP, DBVAR, everything comes under that. CleanVAR is also part of NCBI. All of them are parts of NCBI, but they have different jobs to do. Okay, so they are stored under the same what do you say, web server, hosted by the same web server, but they have the specific job to do. So what are the specific jobs? So basically, gene ontology is world's largest source for your functional gene identification, okay, or finding information on your functions of gene. Then gene expression omnibus. So this is the largest database which stores the raw sequencing data. Let's suppose you are doing sequencing time and again, in the globe, every time sequencing is happening, then a paper got published. So the raw data of that particular sequencing experiment is stored inside this gene expression omnibus, and we can use this particular gene expression omnibus to look for the gene expression quantification, gene expression analysis. I will just demonstrate to you that also. Then comes your DBSNP and DBVAR. So DB is short form of that database for SNP. We pronounce it as SNP, but we write it as SNP, single nucleotide polymorphism. Okay, so a database SNP and database for your variants, that is DBSNP, DBSNP and DBVAR. So these are like catalogs to identify your SNP, or if you want the details of any single nucleotide polymorphism concerning to a gene, so you can find that in the databases like DBSNP and DBVAR. Then comes your CleanVAR. I have already told you what is CleanVAR used for. Basically, if you want to know that a particular variance is having what kind of an effect and how it will lead to a clinical condition, okay, and what is the likely of that to cause a particular disorder or not cause a disorder, for that you can head to CleanVAR. Okay, so then in EBI, so all of this are part of NCBI, then comes on to the all of them are part of NCBI. This is part of EBI, Ensemble. Okay, Ensemble and UCSC genome browser are part of EBI. Okay, so it does the same thing. It is just like how you drove a car, no? So you drive a car. So they are from different companies. Obviously, all of the different cars would be having their own boon and bane. Okay, so likewise, they serve the same purpose. It is just that they would be having their own quality boon and bane that is there. Apart from that, Ensemble is somewhat similar to NCBI's genome browser. Similarly, like UCSC genome browser you have. What is the genome browser used for? So basically a genome browser is majorly used for the first thing that I have just started with. That is, whenever you want to align your query sequence to a reference genome, for that particular purpose, we would be using a genome browser. Okay, and based on the genome browser, we can basically identify what is the variance, and that particular variance is stored in what particular data sets with what particular ID. Okay, so what is the ID? Basically, all of the different data sets would be using a annotation method. Okay, so what is that annotation method? Like DBSNP, for instance, DBSNP, always the annotation will start with RS. Okay, it is called as RSID. Okay, so likewise in your CleanVAR, it will start with VCV. Okay, so that is very specific to a particular variance. Okay, so that is unique. A unique number, you can say a tag. Okay, so unique tag that is being given, so that every time you search via that particular ID, you will be redirected to only and only one particular result. Okay, so that there is no confusion. Okay. So hopefully all of you are getting my point, whatever I'm trying to make. Now coming on to functional and genome, like gene annotation databases. Okay, so for in that we have GeneCart. Okay, so then we have GWAS catalog. I have already described. Okay, so GeneCart can be used for studying again function, expression of genome and association of a particular genome with multiple disorders and like this. Okay. So then comes the second level of omics, that is called as a proteomics. Like obviously, so if certain anomaly happens at the genomic level, that might reciprocate to the proteomic level as well. So if you want to study it at the proteomic level, we will not take up this, just to give you the whole omics category. So if you want to study the variants at the proteomic level, so you can look at the structure of the protein from protein data bank. Okay, so PDB, or a third-party application, or basically it is called a secondary database, that is your UniProt. So what is a secondary database? I have described the primary to you. The secondary databases are curated from primary only, but their purpose is very much okay. Like a primary database would be storing all of the different types of data from the raw experiments, whereas that is not what the secondary database does. The secondary database is very specific to solve a particular kind of problem. Like in this particular case, UniProt. UniProt doesn't generate its own data. It is just like a third-party application which will take the data from AlphaFold, AIML-based AlphaFold nowadays you have. AlphaFold from protein data bank. So it will take all of the data and show it to you in a glance. Okay, so that secondary database does. Okay, so it would be having information from all different data sources, from all different primary sources, so that you can get all of the information in one glance. Okay. Then after the proteome, what we are interested in is the metabolomics, okay, or basically the metabolic cycles, because let's suppose there is a change at the genomic level that is reciprocated to the proteomic level, so the functionality change happens at the proteomic level, and that structural and functional change of the also lead to the change in the metabolome, okay, and the concerning different pathways and cycles that follows. So and the cascade of reaction, so this is the flow of reaction that happens, or basically this is the chronology of thing that happens. Okay, so in order to study a disease, okay, so you can either go from here to genome, or from genome to like trickle down, you can do till here. Okay, so both of the approaches you can use. Okay. So for understanding the proteomics, you have KEGG. So I will demonstrate this one, KEGG. So KEGG stands for basically Kyoto Encyclopedia for Gene and Genomics. Okay. So obviously that is based out of Japan, Kyoto. Okay. You have your string database. So that is for your protein-protein interaction. So all of the proteins that are there, they do interact with themselves, and apart from that, some of the protein might be associated with other proteins. So if you want to work in with one protein, so you have to see that this particular protein is linked to another protein. The whole cascade of things does not get hindered. Okay, so because of that, we study a protein-protein interaction. Okay. So this is what I have already explained to you, what is a primary database and what is a secondary database. So I'm just not shedding light upon this, because we have already discussed on this. Okay. So next thing, so why we are studying all of this? So what is the relevance? Okay, so some of the relevance I have already given you. So the further part of the relevance and how it can help the Ayurveda fraternity or Ayurveda people is the same way, because you are also studying the disease or disorder. Okay, so you can also use from genomics to what do you say, your metabolomics. You can go that like what are the changes that are happening. Okay, so based on Vat, Pitta and Kapha. Okay, so based on that you will be analyzing. But like using this particular databases, what we can do is we can basically search and retrieve. Okay, so let's suppose multiple gene terms we can search using to get to the disorder, what particular disorder it concerns to, or vice versa. I have already told you. Okay, then comes your, like, you want to explore and visualize the results. Would why we say visualize? So certain databases like your genome browser, there you can see physically, you can there is a visual representation that you can see that what is the change, what it should have been and what it is. Okay, so that is the change that you can see, observe. Okay, apart from that, You can also see the representation using gene expression omnibus, where you can see what are the upregulated genes, what are the downregulated genes for a particular disorder or for a particular condition. Okay, let's suppose I'm talking about PNPLA3 gene. Okay, so PNPLA3 gene would be having a positive expression in your liver disorder. Let's suppose we're talking about non-alcoholic fatty liver disease, NAFLD. Okay, so there it would be definitely having a positive regulation. Okay, likewise, so we need to see. So just for an example, I was telling you, and I will show all the demonstration using certain example of gene names. Okay, so in order to contextualize your functions, from like genome to proteome to your genome, and obviously in between the transcriptome also comes in. Okay, so at the end you want to see the functionality of it, because if the structure and the function, okay, so there is a problem with the structure or the function, so that will obviously be translated to the phenotypic changes. So what is a genotypic change and what is a phenotypic change? Hopefully you know. So basically genotypic changes are what we can't see basically. So that is happening in our genome. Okay, but if that particular thing is evident and we can see it with our naked eyes mostly, okay, so that is the resultant of that. Okay, so because of the genetic changes we are having a phenotypic change. Let's suppose someone is having a brown eye, okay, or cat's eye, blue eye, okay, like certain examples of that. Okay, so those are the phenotypic changes likewise. Okay, so that you can infer from the genotypic changes. So then comes your clinical relevance or clinical patient. So because I assume that most of your clinicians, okay, so as most of your clinicians, so this is the main thing that you would be concerned with, I reckon. Okay, for that the databases like ClinVar, DBSNP can help you a lot. And in order to justify your research paper also, how come you can justify your research paper using all of this? Because you see that ClinGen, ClinVar, this particular databases are even recognized by the what do you say, compliance agencies like your US FDA, Food and Drug Administration of India, which gives you the approval for a particular drug. Okay, in India we have CDSCO, in Europe we have EMA. Okay, so likewise, so as like valid evidence, so basically they would consider this particular thing as valid evidence. So let's suppose you are working on a particular disorder, you want to justify that this gene that we have identified, let's suppose like EGFR, okay, gene, or PNPLA3, whatever be the gene, so depending upon the disorder. So this particular gene is having a role in this particular disorder. How can you justify that? Obviously using wet lab experiment, that is there, but which the data which is already there. Okay, so you don't need not to do such a penance of uh, always trying to find out new tricks. So if it is already there, you can simply like use this particular data sets to show them that particular result, and it would be evident enough, and that would be taken into consideration. Okay, then comes on to your drug discovery. So in drug discovery phases, either it be your in silico drug discovery or your wet lab based drug discovery. So we have two approaches basically. So that is there. So in both of them, your GWAS catalog, genome-wide association studies, can help you a lot in order to identify the genetic loci, okay, linked to a particular complex disorder. Complex disorder is what disorder? Basically, it is governed by multiple factors, would be categorized as your complex disorder. Okay, so which is not governed by a single type of change. Let's suppose we are talking about hemophilia and sickle cell anemia. That is not a complex disorder, because only one particular kind of variance will lead to that. Every person that is having sickle cell anemia would be having the same variance. No different variances would be there. But whereas if you're talking about cancer, neurodegenerative disorder, and likewise, in those tangents, you will be having multiple factors governing that. Okay, not only one factor. That is why we need to identify the genetic loci of that in order to do any kind of things that we can do nowadays. We have CRISPR and all if we want to edit thing and likewise. Okay, so that can accelerate the target and like druggable targets it can give us, and the therapeutic interventions we can do using that. Okay, Then comes the personalized medicine. So personalized medicine is basically one person, one drug formula. Basically, will be like personally tailoring a particular kind of a drug for a particular person. Okay, so for that, the genetic makeup of that particular person is absolutely required, and based on that, we can judge that what kind of side effects and those things the particular person might be having, and based on that, we can tailor the drug likewise. Okay, and we can also recommend the lifestyle change regimes and likewise those things. Okay, so for that also, genomics play a bigger part in the diagnostics of that, and in order to manage a particular disorder in a better way. Okay, so that is all from my side as far as the presentation is concerned. So after this, we'll move on to the demonstration. In the meanwhile, if you have any question uh, for the like your initial parts, you may ask, or we will move on to the demonstration. Yes. So shall we begin with the demonstration, or should we wait for some questions?
Participants, if they have any query, they want to clarify anything, so they can please write their query on the chat box. I think Pradyotji, we can move to the next part. If there are any queries, we can take it
Okay. Okay.
later
Thank
also.
you.
Yeah.
Okay. Thank you. So we'll take the questions at the end. Okay. So with that particular note, so I'm moving on to the demonstration part of the things. So side by side, you can also do. And if you are having any problem, you can just let me know at the end of the session. Okay. So we'll try to figure it out. So yeah. So hopefully, yeah. So my new screen is visible to all of you. Can anybody confirm? Okay. So you can move on to your regular search engine. Okay, so Google in this particular case. So you can just straight away go on to the first thing that we are going to look at is NCBI. So because that is the most basic but most powerful tool that is there in bioinformatics. So it is governed by, as you can see, the URL of it. I'm just zooming for the convenience of yours. Okay, so it is your ncbi.nlm, National Library of Medicine. Okay, so under National Institute of Health, government of US. Okay, so we'll just move inside that. So this is the UI you will land up to. Okay, so this is the initial page you will land up to. So now you will be having a drop down menu over here. So of all the databases, so it is having a plethora of databases. Okay, so plethora of databases are there. Okay, so or web servers you can call it as why web server is hosted in web. Okay, so we don't have to download any of the applications, by the way. Okay, so you can directly work in this particular directory in the web. So that is why, what is the advantage of that? So irrespective of whatever system OS operating system you are using, either it a Mac, Ubuntu, Linux, anything you are using, so there is no restriction to that because it is hosted in the web. Okay, so now all of the databases you can see. So whatever we have discussed, you can see dbvar, let me zoom in for you. Okay, so dbvar is there, dbgap, dbvar. Then your SNP you can see. So this SNP is concerning with dbSNP. Okay, and likewise you can also see OMIM that we have discussed. If you go to nucleotide and then search, it will redirect me to GenBank. Okay, so then you have GEO. So GEO also I have told you, gene expression omnibus, and likewise. So we'll see all of them one by one. ClinVar is also there. Okay, so we should basically start with the absolute basic, that is your nucleotide. Okay, so nucleotide. So first things first, that I have told you that we are having the sequence of the things. So how to search for a sequence, let's suppose. So if you have a gene name, so what is the sequence it concerns to? Okay, so let's suppose I go on to nucleotide. So whatever I would be searching, the results would be given to me in GenBank. Okay, so ENPLA3 is the gene. Okay, so that is concerned with your non-alcoholic fatty liver disorder. So nowadays the 60-70% of the population is having NAFLD. Okay, so that is there. So let's search via that gene name. Okay, so here you have the gene. Okay, so the first result that it is giving me is of humans. Okay, so if I go inside that, okay, so here is the search result. Okay, so the full form of that particular gene. Okay, so what it concerns to, what is the symbol of it? Okay, what is the gene ID? So this is a unique gene ID. Okay, so whenever you search via this particular gene ID, you will always land up to PNPLA3 only. Okay, so what is it? Is a protein coding type of a gene, and likewise, so what protein it codes for everything would be shown to you. Apart from that, you also see all of the blue links over here, no? So all of the blue are linkable, what do you say, information. What do I mean by that? If you click on this, so it will redirect you to that particular database on further details of that. Okay, so that we can do. Let's suppose this one. If we are looking for genomic location, so what is the genomic location of this? Which particular chromosome? What is the location of that? If I click on this, so it will redirect me to that. So here you can see. So this is the reference genome to which we are aligning. There you can see. So and this particular PNPLA3 is stored in multiple databases. Here you can see over here. Just give me 1 second. Yeah. So likewise, you can do. Okay, so front and back you can do. So there are a whole lot of things to be explored actually. So I will just point out the important aspect of the things to you. Okay, because in a search result you'll be getting plethora of information. Okay, so you can go and see them one by one. If you are having any issue, you can let us know. Then you have ensemble, you can see the ensemble ID. So this particular thing is also in ensemble that is hosted by your European Bioinformatics Institute. Okay. So this is just kind of a collaborative research. There is no between NCBI and your like EBI and likewise. Okay. So it is a collaborative effort of all of the like database based companies likewise. So if I want to know what disorder it concerns to, I can simply go into OMIM. Here you can see OMIM, the idea is given. So I'm just telling you that what particular thing we can do from a simple gene search. Okay, so I have given you the information that PNPLA3 concerns with NAFLD, but as of now we don't know. Okay, so if I just click on OMIM, now it will give me the information that what it concerns to. Okay, so here you can see what is the gene symbol. So HGNC is basically human, like Hugo Genome Nomenclature Committee. So this is another committee that gives names to the like gene and what particular kind of symbol would be assigned to them, what a particular kind of name would be there. So that is obviously there. So then you have, so this particular thing you can see all of the information are with references it is provided. Okay, so and you can see over here what disorder it concerns to. Okay, so and what is the role of it? PNPLA3 must be having a role. It is not that it is only there for like a disorder. So the role of it is basically fat metabolism. If you read between the lines of here, okay, so you will see that it is used for your what do you say, lipogenesis as well as lipolysis. Okay, basically fat metabolism. You can see expression of fatty acid synthesis and likewise. Okay, so breakdown of fats and synthesis of fat, both it concerns with, and when it malfunctions, then we have the deposition of fat in the liver. So that is why we are having the NAFLD, that is non-alcoholic fatty liver disease. Okay, so that is there. So that is one way. So I will show you multiple ways of searching. Okay, so that is there. Apart from that, if you want to get onto your this thing like instead of a genome browser and all, so if you want to look at the sequence, let's suppose I'm talking about Gales in this particular case, not human. Okay, so I can go to GenBank and see what is the sequence all about. Okay, so this is called as a GenBank format basically. Okay, so this is a GenBank format. Here you can see GenBank is written over here. Okay, and the database is also called GenBank. Okay, so here you have the cytogenetic location and then the like what do you say, your organism name. author information. Okay, so references, author information, journal information, everything is provided over here. Then you come to the origin. No, so in this origin you are having this ATGC sequence, how they are arranged. Okay, so this is the arrangement of the ATGC sequence of that particular location, only that particular location. This is not obviously whole genome. Okay, so only for that particular location which concerns to your PNPLA3 part. Okay, so that is there. So now we can move on to some other databases. Particularly, let's suppose we can move on to your DBSNP. Okay, so for DBSNP, what we need to do? So we just have to select SNP from this particular drop down menu. Okay, so you can see SNP I have selected. You can see SNP. Okay, just to give you a context, I can search using any gene, but just to give you a context, I will be searching using the same gene name. So you can see what is the difference between the results. Okay. So using the same gene, we can basically infer that the results are different and how much different they are. See, now PNPLA3 gene would be having multiple of variances, particularly SNP, that is single nucleotide variances or single nucleotide acid, nucleic acid changes that would be there. Okay, so let's take the first one for instance. So I have already told you that the annotation number or the accession number of DBSNP always starts with RS. So what is the meaning of RS? So RS basically means reference SNP. Okay, and then the serial number, whatever it is submitted. So based on that, so it would be there. And so basically, if a a new entry is just done and it is not yet verified or validated, in that particular case, it will start with SS, submitted SNP. Okay, submitted SNP 1, 2, 3, 4, whatever be the number. Okay, so that is for the context. So let's get on to there. So now once you click on that, so you'll be having the information of what is the variance concerns with. Okay, so I have already told you SNP means one nucleotide acid change would be there. Okay, so what should be there and what it is. Okay, so that is, so in the reference genome, it is always C. See, in the reference genome for a particular location, the gene won't change. The gene will always change in the alternate allele or the query sequence. Okay, and how this particular reference genome is created? So reference genome is just like what you compare to. Just for example, for the people who are not getting this part, let's suppose you do a blood test. Okay, so you are getting your lipid profile done, let's suppose. In that you are getting a cholesterol of 280, let's suppose. So how do I know that particular 280 cholesterol is high or low? I don't have any idea, no. So for that, in the laboratory report, you would be seeing on your right or left, wherever. So it would be written that what is the biological reference to that. Okay, that it should be below 200. Let's suppose the total cholesterol should be below 200. So from that threshold or from that particular thing, you can infer that I am having a higher cholesterol or dyslipidemia likewise. Okay. So those things, how do you know? Because of the reference. Okay, so we always compare a particular thing to the reference or the ideal condition in general words. Okay, so here comes the allele. So what is there? Or if I take 10 people, what 9 people would be having C? Okay, so now we will see what is the alteration. So alteration can be obviously C can't be replaced by C. It can be replaced by A, G, or T. Okay, so that is what we are having over here. So it can be replaced by A, G, and T. But now we have to see in all of the world population studies, what is the frequency of the change? Okay, so I know that it can be converted to T or G, but in what population? What has been studied? You see, in the total world population, it is seen that C changes to T with a frequency of 0.21. Okay, so 0.21 is the frequency of change of C to T. Okay, G 0%, chances are there. Okay, likewise. So likewise, we have to see. And how do we calculate like allelic frequency? Very simple. The percentage, it is just the percentage. Okay, 78.2, like if you convert it to percentage, it would be 78.2. Okay, so likewise, and T, if you convert it to, so it would be 21%, 21.7% to be precise. Okay, likewise. So that is there. Excuse me. So likewise, location you can see. Okay, so in subcontinent population, what is the case? In African population, what is the case? What is the case in the European population? That would give you an idea that if you are studying a particular disorder, so what is the change of like change in the gene, and what is the allelic frequency of that? Okay, here you can see in different studies what is the allelic frequency of T. That keeps on changing, obviously. Okay, so in different studies, so these are individual studies that has happened worldwide. Okay, TopMed is different, and this that likewise. Okay, so you have different studies, twin UK studies, so that is considered twins of UK, United Kingdom. And likewise, HapMap is haplotype mapping. It's a very famous study that has happened in the genomic context. Okay, just like your human genome project. After that, it has happened. So in order to identify the haplotypes. Okay, so that is there. Then you have the different study-based result also written over here. So the reference allele, you can see reference allele frequency and the alternate allele frequency written over here. What is the sample size? That also does. Okay, because when you consider a cohort, obviously you focus on the sample size as well. Okay, and this is the visual representation. What I was telling you, that what is the relevance of a visual representation? This is how if we align it to the genome. So here you can see C to G, you can see now. So C should be in the reference genome, whereas in the alternate we are having a G. And why it is logged and written like this? So this particular variance is having an accession ID by the name of RS738408. Okay, and it is also this particular variance is also there in all of the different databases also, by the way. It is not only in DBSNP. You can see like a reference SNP. So what is the particular variances at different what do you say databases, and likewise you can see, and a single like what is the change that in that particular way it is aligned basically. Okay, so for a beginner you can focus on this particular part. Okay, if you are getting confused. Okay, and this is what is this? This is the length basically. Okay, so obviously there has to be a length in between where the variance would happen. Okay, so here you can see chromosome 22, and this is the location basically, cytogenetic location. Excuse me. So likewise, you can also change the reference genome, by the way. So what is the reference genome? Basically, for the reference genome, we have different versions of reference genome based on the coverage. So we have 3.2 billion base pair. So different coverage would be having, so one is having like 98.7% coverage, one is having 99.8% coverage. Obviously, more coverage, better it is. So likewise, the GRCS 38 is having a better coverage, or it is the latest version that we are having. Prior to that, we had GRCS 37. Okay, and likewise prior to that we have like ng underscore this one. Okay, so you can align to this. What will change? The variance won't change mostly, but it might also change. So mostly your location will change. That is for sure. Okay, so the location that you see, so this particular location that you see now, so this will change. Okay, so that is there. Now coming on to the variance details, if you want to know. So what is the change all about? Then clinical significance, that what disorder it to. Again, same thing. Like if you don't know, okay, so what it causes, see. In CleanVar, you can get the result. Okay, so PNPLA3 I have just searched using. It is giving me NAFLD, no? See, disease name. So by that, I can know that PNPLA3 can cause NAFLD. Okay, what is the clinical significance? This particular accession ID, like this, like G to C, sorry, C to G, pardon. Okay, C to G, whatever is greater is always the reference allele, mind you that. Okay, and whatever is in the smaller side, that is the alternate allele. Okay, so G, like C to G that we have seen, okay, that particular variance leads to a NAFLD condition only, but that is of a benign. Okay, what is a benign type? This particular data is pulled from CleanVar. You can see the CleanVar accession ID. So all of the databases are somewhat connected to each other. We have searched in DBSNP, but when we go to clinical significance, it is taking the data from CleanVar. Okay, because all of them are part of NCBI. they do share their information in between them, so that we can infer the right thing. Okay, so benign, basically, why is that? Let's remove the filter. So benign means the initial stage will cause. Okay, so like for the same gene, you can also have certain variances which will cause like further like pathogenic part of your like NAFLD. That means the later stage of NAFLD, like the fibrosis, then to cirrhosis, then to liver cancer, likewise. Okay. So that is there. So that is the way how you come from gene to disease. Okay. Now there are multiple ways to get from like a pool of gene to disease or disease to gene. Okay, so that we will talk about. So the ClinVar I have already shown you, so I would won't take much of your time in ClinVar. So I will just click on that. I will just show you that how it looks like. Okay, so if you want to know the ClinVar relevance, you can just select a ClinVar over here. Okay, and let's suppose we are searching via EGFR gene. Let's change the gene this time. Okay, EGFR. So you can see the first result that you have got is of pathogenic. So pathogenic means this particular cytogenetic location. Okay, there's some error in loading maybe. There was some server issue maybe. Just 1 second. Okay. So there is some server issue, I reckon, or maybe the browser issue that might occur. But it might work for you if you are changing the browser to Chrome and Firefox. So it might work for you. Okay, so you can change and check it out. Okay, so what we are trying to see in the like summary also we can see. So what is the type of like a variance? It is a copy number gain. Okay, so this is a copy number loss and this is a copy number gain. Okay, so that changes the reading frame of the sequence. Okay, and this particular variance is a pathogenic type. Okay, so if you are studying this particular variance, so this will lead to your like EGFR is mostly related to your lungs based thing. So likewise, so it will be causing a disorder. Okay, although one gene can lead to multiple disorder that is there. Okay, so it is not that one gene variance will lead to only one kind of disorder. It is not that. It might have association with multiple disorders also. Okay, that we can also see for the time being. Okay, so likewise, we can now go to, I think, ClinGen or OMIM. So ClinGen and OMIM, you can search from here also, by the way. So let's select OMIM. Okay, we can search all of the databases one by one in Google also, by the way. Okay, it is not that from here only you have to search. It is just that it is a one-way stop. So that is why I'm like telling you that if you can go via this, so like what would be the search result? Okay, so let's suppose we select OMIM, and this time around, let's do the opposite thing. Okay, we will search via disorder name, and we will land up to what are the genes associated with it. Okay, or which are well studied. Let's talk about type 2 diabetes maybe. Sorry. You can hit on search. Okay. So type 2 diabetes mellitus you need to write. So then it will come. Here you can see. Type 2 diabetes mellitus. Okay. So what is the MIM number? It's just the accession number of your OMIM. Okay, just like we had your like RSID for your DBSNP and likewise. MIM is what accession ID of like your OMIM starts with. Okay. So now you can study the papers where you will be finding the like result of genes. From that, you can also look at the other variances that are there. Okay. gene summary you can go on to. Let's suppose this one. Okay. So here if you read, here also you'll be able to find, but sometimes you will also be finding the gene list in the form of a table. Okay, that is also there. Mapped type 2. Okay, so that is there. These are the references for your understanding. Okay, you can simply go back. We can also look at type 1. Okay. So all of this are clickable links. You can go to PubMed for reading the paper. PubMed is basically to read the papers related to it. Okay. And if you want to like go in details of that, you can simply click on the ID. Already we are in that particular page, so that is why it got read. Okay. Cytogenetic location, this, that, everything is given. Okay. So likewise, you can search. Okay. So yes. Or let's type in once, let's try it out with NAFLD. Generally, you should write the full form. Okay. But you can see it is already taken fatty liver disease susceptibility to 1. Okay. So now you can see mapping non-alcoholic fatty liver disease. Okay. So, and what are the genes concerns to that? You can see PNPLA3 gene, it is saying. I haven't given it anything to say that the PNPLA3 gene is there. Okay. So here you can see PNPLA3 gene is given, even the ID is also given. If I want to know more about PNPLA3, I will click on here. Okay, so the PNPLA3 details will come in. Okay, likewise, you can search. Okay, it is not that only PNPLA3 is responsible, you can also study further. Okay, so there are multiple genes related to it. One of it is PNPLA3, that is the most relevant, so that is why it is mentioned in first. Even the RSID of that is also given, that we have searched using. Okay, so in alcoholic fatty liver, what is the gene doing? Okay, apart from that, these two genes are also involved. Here you can see. Okay, in the inflammation part. Okay, so obviously a disease, if you're talking about the disease etiology, so it will go on to multiple stages, not only at the fat metabolism, but also at the like your inflammation and this, that, immunity based thing, like receptors of the immunity would also be there. And what will happen, it will lead to NASH. Okay, so further progression of that will lead to NASH. Okay, so that is a condition. Like a further condition of NAFLD only. Okay, so then inflammation, this, that. So the whole thing you can study, the concerning genes you can study from here. So I have shown you the both the ways how you can come from gene to disease or disease to gene. Backtracking. Okay. So likewise, you can also start or study with multiple genes or pool of genes that I'm just repeating from that much time. So let's talk about that. Okay, sorry. Yes. So let's get on to the initial page, or here also we can search, by the way. Inside NCBI also you can open GEO. I'm just showing you the other way. So you might be thinking that we cannot search directly. We can search directly also. Okay, like gene expression omnibus if you are writing, although you can do GEO in NCBI, inside NCBI also. Okay, so gene expression omnibus, you can see the second result, gene expression omnibus we will be talking about. Okay, so yeah, here we are. So again, see, it has redirected me to the same NCBI page only. Okay. Now in the accession number, so or the keyword, you can type in like many things. Basically, you can type via the disorder name, or if you are having a GEO accession ID already, so then you can use that. So I am already having one. So if you are not having, you can search by the same, or you can just type in Mycobacterium tuberculosis, which causes TB. Okay, so you'll be redirected to similar result. Okay, so why I'm saying similar? because there would be multiple IDs of your Mycobacterium tuberculosis, or multiple studies concerning to Mycobacterium tuberculosis. It is one of the study. Okay, so ID is GSC 216603. Okay, so just as to give you an insight that what it provides, I have already told you GEO provides us the raw sequenced data. Okay, and what we can use it for? Basically, we can use the raw data for inferring anything or to identify the differentially expressed genes. Okay, those things we can do. So here you can see, so this is the raw. I want to download it. So it is in like a .tar file. So it is zipped basically. So if you download it, it is a like a bigger file format. Okay, so you have to unzip it. So likewise, here you can see the samples. So this is the research paper basically what was published and what they have done. So the summary of that, the important part only is written over here. What you need to see before doing the experiment or basically going on for the quantification of gene or expression of gene. Okay. Then the second thing, you can see the summary of all of the things. Then you have the samples. Okay, so you have always and always whenever you are doing the experiment, you are always having a reference and a test, as you know. Okay, so here you can see one reference, test, experimental, and then test, experimental, test, and likewise it is given. Okay, so 6 samples are there. Okay, 3 are control, 3 are test. Okay, so likewise we need to do, so what we can do over here, there is an exercise that you can do, analyze with GEO to R. So this is a tool basically. Okay, so what it does is, if you don't know coding in R, so for you it is very handy, because using R programming you can come up with the expression quantification, like which particular gene is expressed how, positively expressed or negatively expressed, that you can count. But if you don't know the coding using R, so this particular tool can help you with. Okay, here you don't have to write any code. Okay, just you have to define the groups. Okay, so here all of this 6 are there, so you just need to define groups. Okay, so here is the group. So I would define one group as control. Okay, next group as test. Here you can see the color connotations of there. Okay, so first is control, third control, fifth control. Okay, so just giving an example, you can just see that that which one is controlled and which one is test. Okay, just to give you an example, I'm just running it. Okay, so apart from that, by default, all of the parameters are good enough. Apart from that, if you want to change the p-values and this, that transformations, all of this, that you can do from here, by the way. And if you know R programming, so you can copy this and paste it in your R script. Not required to. Okay, so here I will hit on analyze. Okay, without any coding knowledge, the graphs would be prepared, like based on this particular thing, the graphs would be in front of you. Here you can see. Okay, so here you can see this is called as a volcano plot. Okay, upregulated, downregulated. So the color would be there because of the like Safari doesn't show if you are using a Chrome and all. So you can see the different colors. Okay, so then in the line here you can see this is called as a mean difference plot. Okay, so down regulated and up regulated genes. Okay, that you can see. And if you are not getting, these are just the dots, how can I know that which particular gene is there? So basically this is just a visual representation. Okay, the data is here. This particular data is only plotted in the form of a graph over here. Okay, so don't get confused. So this particular data that you form over here, you can also see the subgraphs over here. So that particular, this particular gene ID, what is the value, what is the sample evaluation, positively regulate control and test, you can see the difference over here. Okay, one by one also you can study. Okay, so one by one you can do, or you can do as a whole like this. Okay, yes, so one by one also you can do, or this is the holistic representation of all of this, all of these things. Okay, so density, even expression density plot, you have expression density plot, UMAP is also there somewhere. Okay, mean variance plot, statistics plot, p-value plot, likewise. Okay, yeah. So that is about your GU. You can also search using Mycobacterium tuberculosis or any other thing also you can search using. Okay, this is just for an example sake I have taken this. Okay, yeah. So let's move on to the next part of the things. Let's get a new tab. Hopefully all of you are following the lecture and doing it side by side if possible. Okay, so the next thing that we will be looking at is gene card. Okay. So in the gene card or basically in ClinGen also, we can search using multiple search terms. Okay. So multiple search terms can be used. Okay, let's suppose I'm taking a particular disorder and taking few of the genes from there. Let's suppose I'm talking about type 2 diabetes mellitus. Okay, so I'll be taking some of the very known gene from there like PPARG. Okay, then like just a space in between, then you can write in all of the gene names A, B, C, C. This is actually a gene name. Okay, so then you have your like G, C, K, R, and likewise. Okay, so there can be many. Okay, so I'm just giving you an example. You can have a pool of gene, a list of genes that you can search using. So if I'm searching using this, here you can see all of the genes result you are getting at once. It is not that one by one also only you can search. Okay. So if I go inside this, okay, so I will see what is the association of the other genes. See, in the metabolism, I can find two of the genes are involved. And in what metabolism? Okay, so here you can see what is the metabolism, what it is affecting. By that you can infer what disorder it is concerning to. Okay, so all of the genes that you have searched using, you will be getting over here. Okay, and if you want to know the metabolic pathways from here, you can click on this. It will be redirected to what particular metabolic pathway it is concerned. It is concerned with. Okay, so here you have the metabolic pathways that it can hinder if any mishappening or basically any malfunction happens in this particular gene. Okay, so that is one way of searching using multiple gene terms, or we can copy the same genes. Okay, use a different software, okay, or data set that is called as a gene ontology. Okay, GO. Okay, so here it will also give me gene enrichment analysis where I can find that all of these genes are concerned with what. Okay, I can give it in a list form. Okay, so here you can see 3 list and I would hit on launch. So click on I'm not a robot and continue. So here it is supported by Panther classification systems basically, and so here you can see the results. Okay, so here you can see the all of the results that are there. Okay, so you can see negative regulation of glucokinase activity, obviously because this gene are concerned with type 2 diabetes, so it would be having the negative regulation of glucokinase activity. Then fat cell differentiation, because in diabetes, as you know, multiple what do you say, your pathways are affected, not only one. Okay, so likewise here you can see all of the pathways that are concerning with this. Okay, and like in Homo sapien, what are the different association that you can get from here? Okay, which particular pathway is positively regulated, negatively regulated? Okay, likewise you can go inside them also, by the way. Like this is PMID is basically PubMed ID, so where you can search for the papers of the same. Okay, apart from that, here you have multiple filters if you want to apply any. Okay, that because geo actually has multiple classes. Okay, so biological function, then your these things are there. Okay, so I will just let you know what are those. Here it is. So anything can be segregated into 3 major things. One is biological process, second one is molecular function, third one is a cellular component. Okay, so whatever gene you are searching using, what biological processes they are involved in, what molecular function they carry out, and what cellular component they are part of. Okay, so likewise you can change the like organism also. Okay, so likewise Homo sapien is selected by default. Example also you can choose, so it will give multiple genes from their side. Okay, so and you can hit on launch and you can see the results. Okay, so it is just to give you a context that how you can search using multiple gene result. Okay, so in multiple gene result, I have shown you gene cut first, second is gene ontology, the third is ClinGen. Okay, so in ClinGen also you can attach multiple genes and see what are the relevance of those. Okay, so like apart from that, you can do a whole lot of things over here also, because it mainly concerns with your clinical relevance, just like clinical variance ClinVar. Likewise, it also concerns with clinical relevance of a particular gene. Okay. Okay, so it is loading. Just give me 1 second. So here they are having the option. So let me click on either of them to give you a context. Okay. So the gene name is there, disorder name is given over here. Okay, so you can search via the disorder name, you will be landing up to gene to disorder, likewise. Okay, and like what are the pathways it taps, this that everything you can find in here. Okay, ClinVar variants also, see there is a relation. ClinGen it is, but ClinVar variants that it concerns with A2ML1. This if you want to search in ClinVar, that also you can redirect yourself to. Okay, so that is there. So I think we have pretty much covered. Only I need to cover FegenI. Okay, so FegenI is there. So in FegenI also nothing different, using the same gene and name, so you can search FegenI. So it is not case sensitive, so you can type in capital letter, small letter, doesn't matter. Okay, so here you can see same thing you can do over here by traits. Acha. So the main advantage in FegenI is that here you are getting multiple options. Actually, you can search by traits also. Okay, physical traits that you can see are symptomatic traits. Via that also you can search. Apart from that, via chromosome or cytogenetic location you can search. Via gene name you can search. Via SNP ID you can search. Okay, so there's those many things you can search using. So see certain examples, gene search result, let's suppose. Okay, so here it is. So I can search multiple genes also, it will take into account, not that. Okay, so it also has a population based thing it is also having, like at what population how much study has been done. Here you can see it also concerns with your type 2 diabetes mellitus. So diabetes mellitus type 2 it is written. Okay, so but as I have already stated, one gene is concerned with only one disorder, it is not like that. So here you can see like in for your disorders also it can have its part. Okay, or like some autoimmunity disorder and this that. Okay, so that you can see from this particular chart. And functional classes, the main thing that is different in here is what are the functionality of this that you can like infer from this particular database, that is your FegenI. Okay, yes, so you can look into all of the data. Okay, so like a whole lot of information you can get from here. Okay, so that is there. So last but not the least is your Jiva's catalog. Okay, so we'll talk about Jiva's catalog and we'll conclude the session over here. After that, if you have any question, you can let me know. So Jiva's catalog. So likewise, whatever studies has happened, pardon. Okay, so whatever studies has happened in the world regarding this any of the disorder concerning genome-wide changes, just like SNP changes, so that you can get in here. Okay, and here you can search via, again you can here also search via traits as well disease. Okay, likewise. So here you can see always and always in any of the bioinformatics database, what is common is the search term. Always it gives you as an example that what are the search term you can use for searching. Okay, like here it has given an example. Disease name you can search using, RSID if you are having, that will also do. Okay, location if you are having cytogenetic location, that will also do. Okay, apart from that you are having the what is it, chromosome number and the location, that will also do. Apart the consent paper if you are having, that will also do. Okay, let's search via an example of Parkinson's disease. Okay, so here you can see how many publications are there. So initially you will get the publication, but if you want to dive deep into the traits, you can go on to the traits. What are the traits concerning to that? Okay. But here you can see all of them T is written. T means these are traits only. These are not publication. If there is a details, it will be shown to you. Okay. So if you go inside this, so you'll be having the association. So that is one thing that no other database will give you. That is your association. Because it is not always in genetics and genomics both that direct inference would be there or direct involvement would be there. There might be some association also, okay, with some other gene that it is influencing. Okay, so that is why you can see the association. What are the different association of this particular gene of causing what kind of a disorder? Okay, here you can see all of the studies. You can, like, if you want to see more, 25 you can do. 25 things will come at once. Okay, so here you can see the reported trait. Okay, and what trait category it is categorized under. So this is, you can see this is common. Why this is common? Because this is a class of traits. Okay, so we have multiple classification of traits, and this particular reported trait falls under this particular what do you say, class of traits. That is young adult onset of Parkinsonism. Okay, so obviously Parkinsonism is a type of a neurodegenerative disorder, as you know. Okay, so where you will be having more of free radical formation and hypoxic condition that happens, and you need more of antioxidant to cope up or deal with that. Okay, so in order to manage that, not in order to eradicate that, because it is a disorder, not to eradicate. So association of that, here you can see all of the associations over here. Apart from that, you can also dive into the studies. So studies are more or less the papers, because all of the studies that has happened in the past would be having a paper also side by side. So the accession ID you can go on to if you want to dive into the details of that particular study, because all of the studies are different, by the way. Here you can see what is the title of the paper, what is the reported trait, those are different. Obviously everything concerns with Parkinsonism or Parkinson's, but different aspects of Parkinson's they are looking into. Okay, so in very much depth you can dive. Okay, so someone is acting on, let's suppose, ubiquitin ligase, okay, of Parkinsonism. Someone is working on the hypoxic conditions concerning to that. Okay, likewise you can go forward. Okay, okay, so that is all from my side as of today. Hopefully you liked the session and you learned something new. So with that note, I would conclude the session over here. And now if you have any question, you can let me know, everyone. Thank you.
Thank you so much, Prodyot. Participants are requested to clarify their doubts by writing their queries in the chat box. Are you able to see, Prodyot? In chat box there is one query.
Yes, sir. Yes. Yeah, you're most welcome, Dr. Sampath. Thanks for your words. How can I computationally identify the most active genes within a specific chromosome within public data bank? Transcription based, you are asking. Transcriptionally active genes. Okay, you just have to click on the exons. Okay, so exons based thing. And can you be a bit more specific about the question? Transcriptionally active
Yeah,
genes with
Dr.
a
Sampath, can you little bit clarify your questions? What exactly you wish to know?
Okay. So I think so. If you go to the Ensemble Genome browser, okay, either you go to the Ensemble Genome browser or you go to the UCSC Genome browser. In both of them, okay, you will be getting that within a rank. I got your point. Okay, I got your point. So within that particular location, because you can see the cytogenetic location over there, and within that particular location, you just have to count upon the exons, because those are the coding parts, as you are telling about the functionally active you want to know. But yeah, so I want to rank the genes which are most active. Okay, but based on what? Geo also we were doing the same thing, na? What you are asking, that like positively regulated is what? So how many genes are positively regulated, and likewise why we were seeing that? In order to know that their activity is governing that particular thing. Okay, so that is what I have given you an example of. Geo would be a thing to do that, or gene ontology can also help you with that. But at metabolomic level, GEO will help, and GEO, gene expression omnibus, will on what you are asking for.
Pradyot, Sangeeta here. I think he's also asking how can we kind of identify the most regulated genes, or the top expressed genes, by computational methods. So I think whatever you have shown, like we have the expression data from different databases. So I think
Yeah,
using R, you can kind of computationally derive it, because
yeah,
expression data can be loaded. Then you have the chromosome information, annotation data. I think we can merge both of them. And then I think all the expression values are given. So just rank it accordingly, and the top rank genes will be the most active genes that are expressed. I think that is basically whatever today's session is kind of the answer to the question, I think.
yes, that is what I have shown you using GEO also. Without using R, you can do that. Like you are using R only, but you don't the code. If you are not proficient in R, in that particular case, you can use GEO to R tool. That uses the R script only, but it is running in the background. Okay, so I can shed more light on that, but for that, a brief of R knowledge is required for like
Yes,
welcome.
yes. Yeah.
Yeah.
Because you have tools like basically libraries like a deployer can help you with that, like if you want to know. How can I classify the gene according to the tissue specific expression retrieval WC hyper? Okay, for that you can go to a AIML tool. I will tell you, because you are asking for a computational part. So you can go for your expecto. Expecto is a SPECTO. Okay, human base expecto. I'm writing it in the chat. Human base expecto. So it does exactly what you are asking, that it gives you the tissue specific expression of the gene in the form of a heat map basically. So like what would be the expression level, how much expression. Obviously it won't be falling in the extreme of the spectrum, it might fall in between also. So heat map would be the right representation to represent that particular thing. Okay. Yeah.
Yeah, participants even they can unmute and directly ask the questions to to Pradyot.
Welcome,
They
Dr.
can unmute also.
Samprat.
If they are comfortable, they can unmute or they can write in the chat box. So there is no more query.
Okay. Okay, so I may conclude then.
Yeah, I think we can conclude it. So
Okay.
thank you so much, Prodyot ji. Thank you so much for exploring this databases. And I think once they have some idea in their mind, they will go through in this database. Then they will get the clear idea and the exact doubt how to do this.
Yes, sir. Yes, right. Yes.
So
Thank you for your kind words. I just wanted to like extend my
yeah.
thank you notes to all the organizers, uh, Bhavana ma'am, and all my fellow colleagues from IGIB. They have helped me a lot in designing all of the things with my busy schedule in the site. So I want to thank each and everyone, and thanks to all the participants as well to bearing to listen to me. So yeah, so that's
Yeah,
all.
thank you so much, Prodyot ji. And one more thing is, Prodyot ji, if any participant would like to contact you, is it possible?
Yes, yes. You can write me a mail.
If you can share your email or the contact
Yes, yes.
in the chat box. So if anyone is having any specific doubt when they are doing the search, they can get guidance from you.
Sure, sure. I'm giving a personal mail ID, not my official one.
Yes.
So I'm giving a personal mail ID so that your query is answered fast. Uh, so here it is. I reckon everyone is able to see that. Can we have a raise of hands if you're able to see that?
Yes, yes, yes. In the chat box, we can see your email ID. So again, I
Thank
will request the participants to get the guidance from Dr. Prodyot. Whenever they are having any doubt, they can directly contact Dr. Prodyot. We have the Google Classroom also. So if you can share some of the study materials with us, so that also we can upload there. So the participants, more than 300 participants are there, so they can go through with these material and they can get better idea.
Yeah, I already have it curated. So I will share with the team in a while.
Or if you have the Google Drive link or anything, that also you can put here.
Yeah, that
Because I have not
Pradyot,
uploaded.
I can upload that in
Or
my
yeah,
chat
or you
also. You
can
can
share
share
with
it with
Dr.
me.
Sangeeta. She will.
Yeah.
She is in the classroom, so
Sure, I will do that. Thank
yeah,
you.
yeah.
Thank you. On behalf of Dr. Bhavana ma'am, I also thank Pradyot for this exhausting and very, lecture filled with clarity, because hands-on lecture is always very helpful for people when they start out. So this was a very useful lecture for us. And tomorrow, I think ma'am will be bridging the genomics and the Ayurveda part. And all the queries that have been coming throughout this lecture as to how we can make use of this information in the field of Ayurveda, in medicine, or in research purpose. I think tomorrow it will be the bridging class. I think the final class also of the series. So I guess tomorrow will be the class will be by Dr. Bhavana. Thank you, Pradyot, for this lecture from our end also.
Thank you everyone. And so I think I will take your leave. Thank you once again. You can always reach out to me in the given mail ID. So with that particular note, bye-bye. Take care.
Thank you so much. Thank you so much to one and all, and especially to Dr. Pradyot ji and Dr. Sangeeta and to